So we're looking at mutations in this activity. And so we want to start with, of course, what are mutations? These are changes in the nucleotide sequence of the DNA. So if it's normally ACC, it may be ACG. It may occur in somatic cells. These are the ones that are not going to be passed on to offspring, so the general cells in your body. Or it can occur in the gametes, the egg and sperm. If it occurs in the gametes, these, of course, will get passed on to the offspring and can lead to changes in the future. So we have a question of whether mutations are actually helpful or harmful, and the reality is it can be both. So they happen all the time. It's a very common thing for mutations to be occurring, and most of them are what we consider neutral. They don't really make a change that we observe. We can have chemicals that can lead to mutations. Ultraviolet radiation can cause mutations. There's a lot of ways mutations can happen beyond the just, oops, there was an error in transcription, translation, DNA replication that didn't get caught. And most mutations that do occur get repaired by enzymes that are constantly checking the DNA long before it ever becomes observable. Now, we know that there are some types of skin cancers, leukemia. There are things that come from mutations in the somatic cells that are considered negative. But there are also the possibility that some mutations might actually improve an organism's survival and be considered beneficial. Interestingly enough, when it comes to malaria, having part of the sickle cell, so like one copy of a sickle cell situation, actually makes you more able to deal with the parasite with malaria than someone who either fully has it or does not have it at all. So sometimes mutations actually do help organisms survive better. So we're going to start with chromosomal mutations. And when we're talking about chromosomal mutations, it might involve changing the structure of the chromosome. So we might have like part of a chromosome missing, or we might lose or gain an entire extra chromosome. So we might have just part of a chromosome affected, or it might be an entire chromosome. So we have 5 key types of chromosomal mutations: deletion, inversion, translocation, non-disjunction, and duplication. So we're going to start with deletion, and this is typically due to breakage. A piece of the chromosome breaks off and it gets lost. So something happens and that piece just kind of disappears. And so when the chromosome basically gets put back together, it's now missing that piece. That's what this picture is showing us. So we have lost information, we have lost genes in this process. In inversion, the chromosome segment is going to break off, and then before it gets put back together, it flips around. So it's backwards. It reattaches, it's still present, but it flips around. And you're thinking, well, it's still there, it doesn't matter. But depending on where the instruction codons to start and stop the process are, you might not actually get the entire gene, or it might try to do it backwards, or it might read a gene that we're not supposed to be reading. So it can still cause problems. Duplication occurs when the sequence is repeated. So something happens, and when it's copying that DNA, it just makes more than one copy. And sometimes you'll see 10 or 20 copies. I mean, it's crazy. Sometimes when this malfunctions, it goes for the gold and malfunctions heavily. Now, there are some of these kind of things that are naturally occurring, and we utilize that to our benefit when we do things like gel electrophoresis. When we do DNA fingerprinting, we actually use these sections where we have repeated DNA sequences. But this is, we're taking one that codes for something specific, and somehow we can end up with two copies of it. That can lead to issues if we make too much of something. Translocation. So this typically we have to have two separate chromosomes that are not considered homologous paired chromosomes. So it's not the two that go together, but two totally different ones. So we might be talking about chromosome 4 and chromosome 18. But what happens is part of one of the chromosomes literally breaks and transfers to another chromosome. So it goes from chromosome 4 onto chromosome 18. So when this happens, obviously we can see that the pieces have literally moved to another place. Now, non-disjunction is probably one of our biggest issues when it comes to chromosomal issues. Because what happens is during meiosis, remember, we have them lined up in pairs, and then the second round they line up single file. But somewhere in this process, things don't separate the way they're supposed to. And so that way, the gamete's going to have either too many or too few chromosomes. Because remember, we're always shooting for example, for humans, we need a total of 46 chromosomes. So when we make a gamete, we want 23, half as many. So that when the egg and sperm meet, they make the 46. And so we might end up with one that has 22 or one that has 24. Normally, it's a slight variation. Very rarely do you see any that have even more non-disjunction with that. But the issue is when we end up with too much or too little, that it doesn't form correctly. Now, I'm going to be honest, many, many times when this happens, we have what is called a miscarriage, which basically is a natural system that's trying to protect against this problem with non-disjunction and this problem with our chromosomal number issues. Okay, so the other side, you can see here on this video, it's showing you kind of running through all the different types of chromosomal mutations and what it's doing to the actual chromosome itself. Okay, so we have the original chromosome. We can have duplication where we have a second copy. Deletion where we take out, notice the D is gone. We can have inversion where they flip a section. And we can have inversion where it basically has reversed and moved it around. So A is still in its same place, but notice that B and C have flipped. So we can flip a whole big sequence, or we can just flip like two pieces. After our chromosomal mutations, and this is actually what we see and tend to focus on mostly, is our gene mutations. So these are a change in specifically the nucleotide sequence of a gene. So if it's supposed to be AACCGCG and we change one of those letters or two of those letters, that's going to be a gene mutation. So it might involve a single nucleotide, and a lot of times it is just a single nucleotide, so we call that a point mutation. But it could be due to some sort of copying error. There could be chemicals that have interfered with the process. There are viruses that will interfere with the process. There's all sorts of things that can cause these errors. So we have point mutations, substitutions, insertions, deletions, and frame shifts that we're going to talk about. And what you're going to find is some of these are overlapping. So a point mutation is a change of a single nucleotide. It could be we add one, we delete one, we substitute one for another. So deletion, insertion, and substitution are all examples that can fall within point mutation. So, for example, sickle cell disease is the result of a nucleotide substitution. A single letter changes. It occurs in the hemoglobin gene and it causes it to change the shape. A frame shift mutation, we're going to insert or delete. So we've changed the number of nucleotides. And if we do one or more, as long as it's not a set of three, it causes, well, even a set of three shifts, but not the same way that one or two does. But it causes a shift in what we call the reading frame, because remember, we always read these in threes. A, U, G, C, C, T. All of these things are always in sets of threes. Three is our magic number. And so if we insert or delete one, it's going to shift it. And so it changes the reading frame. So instead of AUG, we might have AGG. Because we took the U out, it caused everything else to scooch up. And so it changes our sets of three. When this happens, typically our proteins are going to be built incorrectly. So when you're thinking about it, the original, the fat cat ate the wee rat. But if we add one single A, the fat ka, there's and see, all we did is we shifted one over. So here's the T that was originally on the cat, and it pushes everything else. Oh, this one's a deletion. We took out the T, and so we shifted them up. So here's the A for 8. A-T-E. But because we changed that one letter, it causes everything else to shift. So here, the reason it's an issue is we have ATG makes MET, CAA makes glutamine. Well, ATG makes MET, but when we take that G out, all of a sudden now it's AAG, which makes lysine, a totally different substance. And so you can see how that's going to change things behind it. And so here you can see with the video what's happening and shifting as we're making individual changes just at the gene level.